Hello, my name is Caleb Lay, and I am a senior biological sciences and chemistry student at QU. I have been doing chemistry research with Dr. Deskins for 2 years now, and this year we investigated the use of flavonoids as a potential Alzheimer's treatment. In essence, this was a computational study investigating the interactions between flavonoids and the BASE1 enzyme. With more than 3 million cases diagnosed in the United States each year, Alzheimer's disease remains an unrelenting diagnosis that affects many, including family members and caretakers of the affected. While no effective treatment has been shown to reverse the effects of Alzheimer's or prevent its origination, the mechanism behind the disease has been elucidated in part. When amyloid precursor protein begins to break down in the brain as beta amyloid, it is typically eliminated through housekeeping functions. When beta amyloid begins to aggregate, however, these clumps lead to neural breakdown and general dysfunction characteristic to the effects elicited in Alzheimer's disease. Amyloid precursor protein is broken down by the enzyme BASE1. When BASE1 cuts too frequently or imprecisely, many insoluble strands of beta amyloid are produced, resulting in clumps and an increased risk for Alzheimer's disease. Our research team hypothesized that if BASE1 were to cut less frequently, then less beta amyloid plaques would form and less overall neural dysfunction. Through computational protein docking software, we have investigated the theoretical interactions of various plant flavonoids and BASE1 in order to assess if flavonoids may provide interest for further investigation as a drug of choice for treating Alzheimer's disease. Figure 1 illustrates the various flavonoids we used in this study. You can see the general three-ring structure for each flavonoid. In deciding what compounds we wanted to dock with our BASE1 enzyme, we decided upon flavonoids for a couple of reasons. For one, they're small peptide-like. It seemed that most studies in the literature were using small peptide-like molecules instead of larger synthetic ones because they had better inhibition rates. And also, these flavonoids better resembled the reference ligand, which we'll explore in a future slide. And we wanted to use that reference ligand in order to have a kind of a relative value to compare our ligands with. This figure illustrates the BASE1 enzyme alone. It's in a ribbon structure and it was produced by the Argus Lab software. The white arrow at the bottom in the middle is extending into and pointing at the active site of BASE1. It is here where the amyloid precursor protein is cleaved into the nefarious beta amyloid. If we can block this site from the BASE1 interacting with the amyloid precursor protein, then we can limit the amount of beta amyloid that's produced. It is at this site then where we are studying the flavonoids' effects. These protein ligand docking studies were carried out using a crystal structure of the BASE1 enzyme. This structure is available at the protein database website under 6EQM. Water and any co-crystallized ligands were removed from the structure before docking. All compounds were docked with the BASE1 protein using the AutoDock4 software. We used a sphere large enough to accommodate the binding site of the protein in order to allow each ligand to search that area. The different orientations of the ligands were then searched and ranked based on their energy scores. As a check of a docking accuracy, we did a comparison study using the Argus Lab software. Data collected from each docking was tabulated for easy analysis. In the blue columns, you'll see the ligands listed that we used. In the gray columns, you'll see the binding energies in kilocalories per mole, one for AutoDock4 and a confirmation value from Argus Lab. And in the light yellow columns, we see the number of hydrogen bonds. The lowest binding energy values are bolded, as well as the highest number of hydrogen bonds. Here, BUH will serve as our reference ligand, as it was the crystallized ligand already for the 6EQM structure. The lowest value was provided by Argus Lab for epigallocatechin gallate at negative 10.73 kilocalories per mole. Once again, we're looking at the ribbon structure of BASE1, but here we actually see the epigallocatechin gallate docked into the active site as represented as the blue stick model. In contrast, AutoDock4 predicts epicatechin as having the lowest binding energy with negative 6.87 kilocalories per mole. Here we see epicatechin represented as the pink stick model docked into the base 1 enzyme. Note the orientation of the base 1 is different from the other slides. The active site is now at the top of the slide. As seen in Table 1, there is variation between the binding energies of each of the docking programs compared, but within a program, many of the flavonoids actually had similar binding energies. The data for AutoDock 4 show none of the flavonoids had binding energies as low as the binding energy of the crystallized ligand, the BUH, while Argus Lab had its best flavonoid with a lower energy than the crystallized ligand, BUH. Hydrogen bonds were also examined using both programs, and in general, the number of hydrogen bonds had some correlation to binding energies for Argus Lab, but it didn't really seem to be the case for AutoDock 4. While this image is still illustrating epigallocatechin gallate and base 1, it's formatted a little differently. Instead of a whole ribbon structure for base 1, all we see right now are the individual amino acids that hydrogen bond with epigallocatechin gallate. Still in blue, we see pink lines representing the hydrogen bonding, as well as the values in pink right beside those hydrogen bonds. Similarly, we are looking at epicatechin here, hydrogen bonded to the base 1 active site. This image was produced by Autodoc 4. These spheres represent the area of the hydrogen bond. If you look closely, you'll see a green line representing the individual hydrogen bonds. The pink structure is the epicatechin, and you will see the background is the ribbon structure of base 1, and there are individual residues that are associated with the hydrogen bonding, as well as display names for each of these residues. While most of the flavonoids docked in the study did not have lower binding energies than BUH, this actually might have a positive implication for an Alzheimer's drug. Of the base 1 inhibitors that have progressed through clinical trials thus far, they've all been discontinued due to cognitive worsening. Those base 1 inhibitors lowered beta amyloid plaque between 70 to 90%. Having said that, other critical functions of base 1 not linked with beta amyloid production have been elucidated. Inhibition of base 1 reduces plaque, but also negatively impacts important neural pathways. A ligand that inhibits base 1 only mildly may attenuate Alzheimer's disease progression without exhibiting such adverse effects. With this in mind, the data suggests that flavonoids should be further investigated for pharmaceutical use. Given more time, we would like to investigate other flavonoids in addition to variations of the base 1 enzyme. Flavonoids can be extracted and crystallized from plant samples in the lab in order to characterize them. And in vitro studies may then help to reveal preclinical data that's needed to advance flavonoids as a novel pharmaceutical for Alzheimer's disease.